Hello everybody, in this video lecture we will cover the very first chapter, Introduction to Biology. Okay, give me just 1 second, so I'm gonna share the screen with you. I forgot to open my PowerPoint. So this is the PowerPoint that I will use to cover this material. So let's go ahead and begin. Okay, so biology is the scientific study of life. This is what we're gonna do in this class. We're taking biology class, so we're gonna study life and we're gonna use scientific method to do it. But before we begin, we need to answer important question. What exactly is life? For example, are viruses alive? Well, the scientists actually came up with several key characteristics of functions that define living organisms. And they include order, sensitivity or response to stimuli, reproduction, adaptation, growth and development, regulation, homeostasis, and energy processing. Only when viewed together, these 8 characteristics serve to define life. And you see, only when viewed together. So when we look at viruses again, viruses, they have characteristic of living organisms and non-living organisms. So by definition, viruses are not living organisms. Now we will look at all of these characteristics in more details. Order. Order means that all organisms are highly organized structure that consist of one or more cells. And this is called cell theory. So cell theory states that all living organisms, plants, animals, and any other life on our planet, and we assume on any other planet that we will discover in future, all life is made from one or more cells. Cell theory also states that cell is a basic unit of life and all cells came from pre-existing cells. Sensitivity or response to stimuli. Organisms respond to diverse stimuli. Stimulus is change in internal or external environment. So when stuff around you is changing, or maybe your internal parameters change, you will respond. So for example, plants can bend towards the source of light, like shown over here, right? Towards the light, or grows away from the gravity. That is also the stimulus. Tiny bacteria can move toward or away from chemicals that called chemotaxis, or they can move toward or away from light, phototaxis. So those are examples of the sensitivity to the or response to the stimuli. Reproduction. We have sexual and asexual reproduction. Asexual reproduction requires only one parent and offspring are identical copies of that parent. Sexual reproduction requires 2 parents and then offspring are very unique because they have unique combination of DNA from both parents. So in reproduction, DNA containing genes is passed along to an organism offspring. Adaptation. All adaptations enhance the reproductive potential of the individual exhibiting them, including the ability to survive. So we have adaptation to allow us to reproduce better and live longer, and as a result survive, right? Our species will survive if we reproduce and live long enough to pass our genes to the next generations. Growth and development, as shows over here, organisms grow and develop according to specific instructions coded by their genes. Regulation. Even the smallest organisms are complex and require multiple regulatory mechanisms to coordinate internal functions, such as transport of nutrients, response to stimuli, coping with environmental stresses. Well, means that everything in your body needs to be regulated, right? The amount of oxygen in your blood, the amount of urine produced, how your muscles contract, what hormones are secreted in your blood, right? Amount of sugar, glucose level in your blood, that's everything needs to be regulated. Another property of life is homeostasis. Homeostasis is related to this regulation because you regulate your internal environment to keep it in balance, to keep it in homeostasis. Homeostasis means steady state, the ability of an organism to maintain constant internal condition. For example, shown over here, your body temperature. When your body temperature increases, you do have mechanisms that return it back to normal, and the same happens when your body temperature decreases. So that's homeostasis. Energy processing. All organisms use a source of energy for their metabolic activities. Some organisms capture energy from the sun and convert it into chemical energy. We call them autotrophs. And some organisms use chemical energy from molecules they take in, that means from their food, and we call them heterotrophs, and human are heterotrophs. Okay, so just very quickly, I just go back over here and I'm gonna remind you that we just covered these properties of life, these 8 characteristics. And if we have some structure, right, that has all of these characteristics, we call it living organism. For example, let's say if I'm talking about stomach, human stomach. Is the human stomach a living organism? It is not, because, for example, it cannot reproduce, right? It cannot reproduce. It cannot adapt. So your stomach, even it's made from cells, it's not a living organism, it's a part of living organism. And again, because it does not shows all these characteristics of functions. And now we're gonna talk about what is scientific study. Oh, okay, so I'll go over here. So there are 2 main scientific approaches. Discovery science is mostly about describing nature. That's why we call it discovery science. And hypothesis-driven science is mostly about explaining nature. And this one includes experiment. So discovery science collects verifiable observation and measurements. So those are data for discovery science. In biology, discovery science enables us to describe life at its many levels, from ecosystem down to cells and molecules. For example, if you have a light microscope and you're looking at a drop of water from a local lake, so you're making your observation about type of living organisms that inhabit that lake, right? So you're practicing discovery science. Right, and this discovery science stimulates you to ask questions and seek explanations. And to answer those questions, we use hypothesis-driven science. And hypothesis-driven science includes inquiry called scientific method that consists of series of steps that provide a loose guideline for scientific investigation. So scientific method include precise steps that we need to do in order to support or deny our hypothesis, right? And we will look at these steps. So most modern scientific investigation can be described as hypothesis-driven science. Hypothesis is a tentative answer to a question, explanation on trial. And this is very important. Hypothesis cannot be proved true. Hypothesis can be only supported or disproved. So you can disprove hypothesis, you cannot prove it. The good hypothesis always written in this form, if, then. And sometimes they add due to. And what you put over here and over here, those are variables. So this is independent variables, something that you will change. And this over here, result would be your dependent variable. This will be outcome of your experiment. So here's the steps of scientific method. Observation and research. Make observation and research your topic of interest. Problem question, develop a question or problems that can be solved through experimentation. Problem question, this is purpose of the experiment. Right, so it's not just any question. You cannot ask question like, I wonder, why am I doing this experiment, right? This is not scientific question. This kind of like another word for problem equation would be purpose of the experiment. Formulate a hypothesis. That means predict a possible answer to this question that you just developed. Example would be this, for example, right? If soil temperatures rise, then plant growth will increase. So you see, if we do something, if we change something, then something happens. And soil temperature will be your independent variable. And plant growth, 20 centimeters, 30 centimeters, 50 centimeters, that will be your dependent variables. Actually, to understand experiment, you need to understand what are your dependent and independent variables. After you formulate a hypothesis, you develop your experiment and you follow a procedure. You include a detailed material list. We need procedure and material list so other scientists can repeat your experiment. After experiment is done, collect and analyze results, modify the procedure if needed, confirm the results by retesting, include tables, graph, and photographs. So this is where you will include your data. In the results, you don't need to give any explanation. You do not mention any hypothesis. You just state what you measured, what you saw, what you recorded. And you also need to analyze kind of like patterns and trends, but you don't give any conclusion in the results because you have a conclusion section, right? In the conclusion, you need to include a statement that you accept or reject the hypothesis. And in the conclusion now, you can explain why you get these results and so on. So in the results, it pretty much just your data and some patterns that you see, right? So the highest temperature, when, for example, if you go back to our temperature over here, if soil temperature. So in the result, you can say when temperature increased by I don't know 5 degrees of Celsius, the average height also increased by 5 centimeters. So you see you analyzing your numbers, right? Or you can say the highest increase in plant's height was achieved at temperature of 27 degrees. Or you can also say that we saw the correlation with increase of temperature and increase of the height of the plant up to a certain point, and then height of the plant decreased again, right? So you describing what exactly we can see from these numbers, right, that you collected, from the data that you collected. That's for results. Now, in the conclusion, again, you're gonna say if you accept or reject the hypothesis, you make recommendations for further study and possible improvements to the procedure, and then communicate the results. That's important section. You make your data available for other scientists. First, they might wanna repeat your experiment to support or deny your hypothesis, or they can build up their work based on whatever you already discovered. Part of a controlled experiment include independent variables or manipulated variable is a factor that's intentionally varied by the experimenter. Dependent variable or responding variable is a factor that may change as a result of changes made in the independent variable. Control group is exposed to the same condition as experimental group, except of this independent variable, the variable being tested, and constants or standardized variables. In the experiment are all the factors that the experimenter attempts to keep the same. So here, for example, we have control group, a pot without water, an experimental group, a pot with the water. And what you're doing here, you research how your independent variable, amount of water, affects the number of seeds that sprout. So number of seeds that sprout that you will count, this is your data that you will collect, right? And this will be your dependent variables. Your dependent variables are your data. But why do you have 9 out of 10 seeds sprout over here? Because you use your independent variable of water, right? So here's your independent water, right? Your dependent fraction of seeds that sprout. You have your experimental group, and you have your control group that exposed to the same variables except of water, except of independent variables. Your constants, your constants will include identical pots, identical seeds, the same amount of light, right? And same amount of time that you gave these 2 pots for seeds to grow, right? So everything else except of water will be your constants, right? Water will be independent variable, number of seeds that sprout will be dependent variable, experimental group, this is the one that you apply your independent variable, control group, you do not apply independent variable. So now what is scientific theory and how it is different from a hypothesis? Scientific theory is much broader in scope than a hypothesis. Theories only become widely accepted in science if they are supported by accumulation of extensive and varied evidence. So we have our experiments, right? Through the experiments, we perform experiment, we can create and modify the theory, and then we use theory to make more predictions. And then we research this prediction. So we design experiment to test this prediction. And again, we're supporting the theories. So scientific theories are not the only way of knowing nature. Science, religion, art are very different ways of trying to make sense of nature. So you can see here on this pie diagram and how they all overlap except religion and science. Religion and science only touch each other. There is no overlapping between religion and science. That means science has nothing to say about religion. So science, however, has 2 key features that distinguish it from other forms of inquiry. So we just said that science is not the only one way of knowing nature, but science has 2 key features that distinguish it from any other form of inquiry. Science depends on observations and measurements that others can verify and requires that ideas or hypotheses are testable by experiments that others can repeat. So science depends on everything that can be verified or repeated by others. So here's the culture of science. Science uses scientific method. Ideas must be tested. Science can only look at natural world, and there is no absolutes. In science, scientific laws are not considered absolute truths. In fact, the core philosophy of the scientific method is that no scientific fact is to be considered an absolute truth. Isn't it beautiful? We're talking about no scientific fact. See the word fact, and this, even the law of fact in science is not absolute truth. That makes scientists very humble people, because they always, in the back of their head, they always remember there is no such thing as absolute truth. Everything can be disproved at some point of time. And I also want to look at this, that science can only look at natural world. So what is natural world? In the language of science, the term natural refers to any element of the physical universe. In other words, everything that is made from elements, and you find those elements in periodic table. If something is made from those elements, it's a part of natural world. Natural is often identified by testability. Natural things behave in a predictable way, though we may not yet fully understand them, which have observable outcomes. This predictability means that we can test hypotheses about natural entities by making observations. Ghosts, for example, are supernatural entities without a basis in the physical universe, and so are not subject to the laws of that universe. Hence, ghosts are outside of purview of science, and we cannot study their existence or lack of it with the tools of science. And the last part of this chapter is about levels of organization. So if you take a biosphere, right, this is everything that supports life on our planet, and you start breaking it into smaller and smaller and smaller and smaller units, you will get to the atomic level of organization. So we're gonna start with atoms. Atom is the basic unit of matter. Then atoms make molecules. Many molecules became large molecules. We call them macromolecules, organic molecules. These organic molecules make organelles. Organelles make cells. Cells make tissues. Tissue make organs and organ systems. Organisms make population, communities. Communities make ecosystems, and ecosystem make the biosphere. And living organism must be classified. All life on this planet is divided into 3 large groups. We call them domains. And those domains include eukarya, archaea, and bacteria. So that's a bacteria domain, archaea, and eukarya. And we will talk about these domains a little bit later. But then those domains are divided into kingdoms. So bacteria and archaea, they do not have any kingdoms. Eukarya used to be these fungi, plants, animals, rodents. This group does not exist anymore. If you look at cell type, some cells are prokaryotic. Prokaryotic cells means those cells do not have nucleus, but they do have DNA. In domain eukarya, cells are eukaryotic. Those cells have nucleus and other membrane-bound organelles. Cell number, bacteria and archaea are made from 1 cell. We call them unicellular organism. Eukarya, most of eukarya are multicellular. bodies can be both. And here's our energy. It can be autotrophic, heterotrophic, right? So that's some characteristic used in classification of organisms. And in 19th century, scientists named Carl Linnaeus first proposed organizing the known species of organisms into hierarchical taxonomy. When we cover evolution unit, we will talk about these groups. But domains are the larger groups. They divided into kingdoms, then in phylum, class, order, family, genus, and species. So species are the smallest group. They very specific and very unique. So organisms from different species should not interbreed. They should not produce viable offspring. However, some exceptions happen, of course, because dog and wolves sometimes can interbreed. That just means they're very close species, right? So here they even put them in the same, pretty much, species. I think that's it. So this is the last slide. And as I said, this is introduction to biology. And what we cover very briefly, like domains and kingdoms and cell types and this taxonomy, everything will be covered in our further lectures. So that's it for now. Thank you for watching, and I hope it was helpful.